Okay, so welcome to this next video in which we are discussing quorum signaling. So we have discussed that these molecules, N-acyl homoserine lactones, are often used by bacteria as quorum sensing molecules. So we're now discussing how a bacterium such as E. coli transports the N-acyl homoserine lactone out of its cytoplasm and into the extracellular space. Okay, so we've seen the two examples of these massive great transporters, ACR-AB-Tol-C, which spans both the inner membrane and the outer membrane of Escherichia coli, and will transport N-acyl homoserine lactones out of the cytoplasm and into the extracellular space. And it's an example of an RND transporter, or a resistance nodulation cell division transporter, which means that it uses secondary active transport to move the N-acyl homoserine lactone out. So it takes advantage of the proton gradient across the membrane of across the inner membrane of the E. coli cell. Now, E. coli is what's known as a facultative anaerobe. Okay, a facultative anaerobe. It usually lives in the intestine, which means that which is an anaerobic environment, i.e. very low levels of oxygen. So its usual metabolism is anaerobic, so it doesn't use oxygen to produce ATP. That means that it doesn't have an electron transport chain that is usually functioning. So, instead, the way that it builds up this proton gradient across its inner membrane is by proton pumps. So it generates its energy by other means, and then when it has this ATP, it will then use the ATP to drive the movement of protons across this membrane, basically. So to move protons from the cytoplasm into the periplasmic space, and it will couple that movement to the hydrolysis of ATP. So it will actually put energy in, basically, to establish this proton gradient, so that then protons can move back down their electrochemical gradient, and that movement can be coupled to the movement of other molecules such as N-acyl homoserine lactones out of the cell. And both NOR-E and ACR-AB-Tol-C both use this principle. So, this is how we extrude these N-acyl homoserine lactone molecules into the extracellular space around the bacteria. Now, when just one bacterium is living on its own, when it secretes these quorum sensors, the quorum sensor will just diffuse off, and it will have a tiny little concentration, basically. Okay, but as I say, if they are living in very close proximity, okay, so if the population now increases, so let's draw more of them, then there will be absolutely loads of these E. coli bacteria producing this quorum sensing molecule, these N-acyl homoserine lactone molecules. So basically the concentration will go up and up and up. Now this indicates to the bacteria that the population is very dense, basically. So the bacteria sense the high levels of N-acyl homoserine lactone in their extracellular fluid, and that causes them to stop dividing, basically. Okay, so they go into the stationary phase. Now, that's what causes that to result in that initial experiment. That's why they stop dividing, because once the density reaches a certain point, they say, we don't need density to go any higher, we'll stop dividing now. And this concentration of quorum sensing molecules is what tells them that their population is at that high enough level that they should go into the stationary phase now. Okay, now there are two quite interesting experiments you can do. What you can do is you can knock these transporters out in the E. coli. So you can genetically modify the E. coli so that they don't produce an ACR-AB-Tol-C transporter and don't produce a NOR-E transporter. Now if you knock those out, what's going to happen? They're no longer going to be able to they're no longer going to be able to transport N-acyl homoserine lactones out of their cytoplasm. So the amount of N-acyl homoserine lactones that these bacteria are secreting goes down hugely. Okay, so for this same population density that we have here, the density of the NaCl homoserine lactone molecules will be much lower. And you might say, well, why doesn't it go down to 0? Well, because this isn't the only way the NaCl homoserine lactones can get out of the cell cytoplasm, but it's one of the ways. So knocking them out doesn't completely abolish the secretion of NaCl homoserine lactones, but it does reduce it. So for the same population density, you're going to have a much lower density of NaCl homoserine lactones. Okay, and that means that the bacteria will not stop dividing yet. So instead, what they'll do is they'll need to get to a much higher density before, at this impaired level of NaCl homoserine lactone secretion, they will actually have a high enough density of the molecule that they will stop dividing. So the density that they'll be at when they undergo when they go into the stationary phase will be much higher, basically. So basically, by knocking out the ACRAB, TOL-C, and NOR-E transporters, you push back the density at which they will go into the stationary phase, basically, and it's because you've impaired the secretion of this quorum molecule, quorum sensing molecule. Okay, in contrast, if you upregulate the expression of these ACRAB, TOL-C, and NOR-E transporters, then they will all transport more NaCl homoserine lactone molecules out of their cytoplasm. So each one will secrete more NaCl homoserine lactone molecules, and at a certain density, the density of NaCl homoserine lactones in these bacteria with this upregulated expression of ACRAB, TOL-C, and NOR-E will be greater, basically. So they will get to the necessary concentration of NaCl homoserine lactones at a lower population density of the bacteria. So you'll need a lower density of bacteria in order to produce the right density of NaCl homoserine lactones that they should go into, that tells the bacteria to go into the stationary phase. So if you upregulate them, what happens is the bacteria go into the stationary phase much earlier, basically, when the population density is much lower than it would be if they hadn't had the ACRAB, ACRAB, TOL-C, and then NOR-E transporters upregulated. So those are some nice experiments which confirm our theory.